Welcome to our deep dive into the female reproductive system, a fascinating and complex part of human anatomy. In this video, we'll explore the organs, processes, and hormonal regulation that make the female reproductive system unique. From the ovaries to the uterus and the intricate cycles that govern fertility, you'll gain a comprehensive understanding of how these systems work together. We'll also touch on developmental aspects, clinical concerns, and the role of hormones in female health. Let's begin our journey by setting the stage for what makes the female reproductive system so remarkable. Reproductive role of the female is more complex than that of the male, primarily due to the demands of pregnancy. The female gonads, known as ovaries, are responsible for producing gametes called ova and secreting essential sex hormones like estrogen and progesterone. These hormones regulate not only reproductive functions, but also influence secondary sex characteristics and overall health. The internal genitalia, including the ovaries, uterine tubes, uterus, and vagina, are located within the pelvic cavity, while the external genitalia are visible outside the body. Understanding these structures is crucial for appreciating the intricacies of female reproduction. Let's take a closer look at the organs of the female reproductive system. The midsagittal section reveals the spatial arrangement of the ovaries, uterine tubes, uterus, and vagina. Each organ plays a specific role in the reproductive process, from gamete production to nurturing a developing embryo. The ovaries are the site of oogenesis, while the uterus provides the environment for implantation and growth. The vagina serves as the passageway for childbirth and menstrual flow. This anatomical overview sets the foundation for understanding how these organs interact during the reproductive cycle. Internal female reproductive organs are intricately designed to support the processes of ovulation, fertilization, and pregnancy. The ovaries contain follicles at various stages of development, each housing an oocyte. The uterine tubes facilitate the movement of ova toward the uterus, where fertilization may occur. The uterus itself is composed of layers, including the endometrium, which undergoes cyclical changes in response to hormonal signal. These organs work in harmony to ensure reproductive success and maintain overall health. Photomicrograph of a mammalian ovary reveals the microscopic structure of follicles and oocytes. This view highlights the cellular complexity and the stages of oocyte development, from primordial to mature follicles. The ovary is not only a site for gamete production but also for hormone secretion, which influences the menstrual cycle and secondary sex characteristics. Observing the ovary at this level helps us appreciate the biological intricacies involved in female reproduction. Endometrium, the inner lining of the uterus, is essential for implantation and pregnancy. Its blood supply is rich and dynamic, adapting to hormonal changes throughout the menstrual cycle. During the proliferative phase, the endometrium thickens and prepares for potential embryo implantation. If fertilization does not occur, the functional layer is shed during menstruation. Understanding the endometrium's structure and blood supply is key to grasping the physiological changes that occur each month. Endometrium undergoes significant changes during the menstrual cycle, influenced by estrogen and progesterone. Spiral arteries supply nutrients and oxygen, supporting the growth of the functional layer. These changes are crucial for creating an optimal environment for embryo implantation. If pregnancy does not occur, hormonal shifts cause the endometrial layer to break down and be expelled, marking the start of a new cycle. This cyclical process is central to female reproductive health. External genitalia, collectively known as the vulva, include the labia majora, labia minora, clitoris, and vestibule. These structures protect internal organs and play a role in sexual arousal and intercourse. The vulva is rich in nerve endings and blood vessels, contributing to sensitivity and lubrication. Understanding the anatomy of the external genitalia is important for recognizing their functions in both reproductive and sexual health. Closer look at the vulva reveals its detailed anatomy. The labia majora and minora encircle the vestibule, which contains the openings for the urethra and vagina. The clitoris, a highly sensitive organ, is central to female sexual response. These external structures are not only protective but also facilitate sexual pleasure and reproductive functions. Their health and integrity are vital for overall well-being. Oogenesis is the process by which female gametes or ova are produced. It begins during the fetal period, with oogonia dividing by mitosis to form primary oocytes. These oocytes undergo meiosis, but the process is arrested at specific stages until puberty or fertilization. Oogenesis is distinct from spermatogenesis, with unique timing and outcomes. At birth, females have a lifetime supply of primary oocytes, setting the stage for future reproductive cycle. There are three key differences between oogenesis and spermatogenesis. First, primary oocytes are produced only during fetal development. Second, meiosis in primary oocytes is arrested in late prophase I and resumes years later if at all. Third, meiosis in secondary oocytes is arrested in metaphase II and only completes if fertilization occurs. These differences highlight the unique nature of female gamete production and its regulation throughout life. Ovarian follicle is the functional unit of the ovary, enclosing a single oocyte surrounded by pregranulosa or granulosa cells. Follicles progress through stages of development, starting as primordial follicles with a single layer of cells. As they mature, the cell layers increase, and the follicle becomes primary, secondary, and eventually vesicular. This relationship is crucial for understanding how oocytes are nurtured and prepared for ovulation. Follicles develop through several stages. Primary follicles have a single layer of cubital cells, while secondary follicles have multiple layers of granulosa cells. Vesicular follicles, also known as antral or tertiary follicles, contain a fluid-filled cavity called an antrum. Before ovulation, the primary oocyte inside the vesicular follicle resumes meiosis and becomes a secondary oocyte. This maturation process is essential for successful ovulation and fertilization. Follicles have two possible fates, atresia or ovulation. Atresia involves apoptosis or programmed cell death of the oocyte and surrounding cells. Ovulation occurs when hormonal signals, especially FSH, activate a select group of primary oocytes each month. One dominant follicle is chosen to mature and release an oocyte while the others undergo atresia. This selection process is vital for reproductive health and efficiency. The dominant follicle resumes meiosis just before ovulation. Two haploid cells are produced, the first polar body, which is small and almost devoid of cytoplasm, and the secondary oocyte, which retains most of the cytoplasm and organelles. The secondary oocyte is the cell that will be ovulated and potentially fertilized. This division ensures that the ovum has enough resources to support early embryonic development. Secondary oocyte arrests in metaphase II and is the cell that will be ovulated. If it is not penetrated by sperm, it deteriorates. If fertilization occurs, the secondary oocyte quickly completes meiosis II, yielding an ovum and a second polar body. The ovum contains enough cytoplasm to nourish the fertilized egg during its journey to the uterus, while the polar body degenerates and dies. Common question regarding oogenesis is which statement is incorrect. Production of primary oocytes occurs only in the fetus. Meiosis in primary oocytes is arrested in late prophase I. Meiosis in secondary oocytes is arrested in metaphase II. Or at birth, females are presumed to have a lifetime supply of secondary oocytes. The correct answer is that females have a lifetime supply of primary oocytes, not secondary oocytes. This distinction is important for understanding female fertility. Oogenesis and spermatogenesis differ in several ways. Oogenesis produces one viable haploid ovum and 2 to 3 polar bodies, while spermatogenesis produces 4 viable sperm. Oogenesis begins in fetal life and ends at menopause, whereas spermatogenesis starts at puberty and continues into old age. Error rates also differ, with oogenesis having a higher error rate of 20% compared to 3–4% in spermatogenesis. These differences reflect the unique challenges and processes of female gamete production. Follicle maturation occurs in two phases. The gonadotropin-dependent preantral phase involves paracrine signals within the follicle, while the antral phase is stimulated by FSH and LH. During the antral phase, a dominant follicle is selected, and the primary oocyte resumes meiosis I. This maturation process can take nearly a year, highlighting the complexity and timing involved in preparing an oocyte for ovulation. Transition from primordial to primary follicle involves squamous-like cells surrounding the primary oocyte becoming cuboidal, and the oocyte enlarges. This process can take about 1 year. The oocyte secretes glycoprotein-rich substances that form the zona pellucida, encapsulating the oocyte. This step is crucial for protecting the oocyte and preparing it for further maturation. As the primary follicle becomes a secondary follicle, pregranulosa cells proliferate, forming a stratified cuboidal epithelium around the oocyte. When more than one layer of cells is present, they are called granulosa cells. The follicle is now termed secondary, and connective tissue in granulosa cells condense to form theca folliculi. Thecal cells secrete hormones in response to LH, supporting further follicle development. Secondary follicle stage ends when liquid accumulates between granulosa cells, forming an early vesicular follicle. The antrum, a large cavity, is formed as all fluid coalesces. This stage marks the readiness of the follicle for ovulation, as the oocyte becomes isolated with its surrounding granulosa cells, known as the corona radiata. Corona radiata, consisting of granulosa cells surrounding the oocyte, sits on a stalk within the vesicular follicle. When the follicle reaches full size, it bulges from the ovary surface and is ready to be ovulated. This process ensures that the oocyte is well protected and supported as it prepares for release and potential fertilization. After ovulation, the ruptured follicle transforms into a glandular structure called the corpus luteum. This structure is essential for hormone production, particularly progesterone, which supports the uterine lining and maintains pregnancy if fertilization occurs. The transformation from follicle to corpus luteum is a key event in the ovarian cycle. Ovarian cycle is a monthly series of events associated with the maturation of an egg. It consists of 2 consecutive phases, the follicular phase, which involves vesicular follicle growth, and the luteal phase, which is marked by corpus luteum activity. Ovulation occurs mid-cycle, and while the follicular phase can vary in length, the luteal phase is consistently 14 days from ovulation to the end of the cycle. During the follicular phase, several vesicular follicles become sensitive to FSH and are stimulated to grow. One dominant follicle outcompetes the others and continues to mature, while the non-dominant follicles undergo atresia. FSH levels drop around the middle of the phase, ensuring that only the dominant follicle progresses to ovulation. The primary oocyte of the dominant follicle completes meiosis I to form a secondary oocyte and the first polar body. Granulosa cells then send signals to the oocyte, causing it to stop meiosis until fertilization occurs. This precise regulation ensures that the oocyte is ready for fertilization at the optimal time. Ovulation occurs when the ballooning ovary wall ruptures, expelling the secondary oocyte with its corona radiata into the peritoneal cavity. Some women experience a twinge of pain known as Mittelschmerz during ovulation. Occasionally, more than one oocyte is released, leading to fraternal twins if both are fertilized. Identical twins result from the fertilization of a single oocyte followed by separation of daughter cells. After ovulation, the ruptured follicle collapses and the antrum fills with clotted blood, forming the corpus hemorrhagicum. This structure is eventually absorbed, and the remaining granulosa and thecal cells enlarge to form the corpus luteum. The corpus luteum secretes progesterone and some estrogen, which are essential for maintaining the uterine lining and supporting early pregnancy. Pregnancy does not occur, the corpus luteum degenerates into a scar-like structure called corpus albicans within 10 days. If pregnancy occurs, the corpus luteum continues to produce hormones that sustain the pregnancy until the placenta takes over at about 3 months. This transition is crucial for supporting the developing embryo during the first trimester. Hormonal regulation of the ovarian cycle begins before puberty, when ovaries secrete small amounts of estrogen, inhibiting the release of GnRH from the hypothalamus. As puberty approaches, leptin levels rise, making the hypothalamus less sensitive to estrogen. This allows GnRH to be released, stimulating FSH and LH production, which act on the ovaries to initiate the adult cyclic pattern and menarche. During the ovarian cycle, GnRH stimulates the secretion of FSH and LH. These hormones promote follicle growth, maturation, and the secretion of sex hormones. FSH stimulates granulosa cells to release estrogen, while LH prompts Theckel cells to produce androgens, which granulosa cells convert to estrogens. This hormonal interplay is essential for coordinating the events of the ovarian cycle. Negative feedback mechanisms inhibit gonadotropin release. Increasing plasma estrogen levels exert negative feedback on FSH and LH release, while inhibin from granulosa cells further inhibits FSH. Only the dominant follicle can withstand this dip in FSH, ensuring that it continues to mature while other follicles deteriorate. This regulation maintains balance in the ovarian cycle. Positive feedback stimulates gonadotropin release as estrogen levels continue to rise due to the dominant follicle. When estrogen reaches a critical high value, a brief positive feedback occurs on the brain and anterior pituitary, triggering an LH surge. This surge is essential for ovulation and the completion of meiosis in the oocyte. LH surge triggers ovulation and the formation of the corpus luteum. High estrogen levels prompt the release of stored LH and some FSH by the anterior pituitary at midcycle. This surge causes the primary oocyte to complete meiosis I, becoming a secondary oocyte, which then enters meiosis II and arrests at metaphase II until fertilization. LH also stimulates events that lead to ovulation, such as increasing local vascular permeability and triggering an inflammatory response. Metalloproteinase enzymes weaken the ovarian wall, causing it to thin, bulge, and rupture. The oocyte with its corona radiata exits, completing the process of ovulation and entering the peritoneal cavity. Shortly after ovulation, estrogen levels decline, and LH transforms the ruptured follicle into the corpus luteum. LH stimulates the corpus luteum to secrete progesterone and some estrogen almost immediately. Progesterone helps maintain the stratum functionalis of the uterus and supports pregnancy if it occurs. Negative feedback from rising plasma progesterone and estrogen levels inhibits LH and FSH release. Inhibin from the corpus luteum and granulosa cells enhances this inhibitory effect. Declining LH ends luteal activity and inhibits further follicle development, ensuring the cycle progresses in a controlled manner. Fertilization does not occur, the corpus luteum degenerates as LH levels fall, causing a sharp decrease in estrogen and progesterone. This ends the blockage of FSH and LH secretion, allowing the cycle to start anew. The oocyte is actually activated 12 months prior to ovulation but matures 14 days before ovulation, highlighting the long-term planning involved in female reproduction. Uterine or menstrual cycle is a cyclic series of changes in the endometrium that occur in response to fluctuating ovarian hormone levels. It consists of 3 phases: the menstrual phase days 1-5, the proliferative phase days 6-14, and the secretory phase days 15-28. Each phase is characterized by specific hormonal and structural changes that prepare the uterus for potential pregnancy. During the menstrual phase, ovarian hormones are at their lowest levels and gonadotropin levels begin to rise. The stratum functionalis detaches from the uterine wall and is shed, resulting in menstrual flow that lasts 3-5 days. By day 5, growing ovarian follicles start to produce more estrogen, signaling the end of menstruation and the start of a new cycle. Proliferative phase is marked by rising estrogen levels, which prompt the generation of a new stratum functionalis layer. As the layer thickens, glands enlarge and spiral arteries increase in number. Estrogen also increases the synthesis of progesterone receptors in the endometrium and thin cervical mucus to facilitate sperm passage. Ovulation occurs at the end of this phase. Secretory phase is the most consistent in duration and prepares the endometrium for embryo implantation. Rising progesterone levels from the corpus luteum prompt the functional layer to become a secretory mucosa. Endometrial glands enlarge and secrete nutrients, while thickened mucus forms a cervical plug to block entry of sperm pathogens or debris. If fertilization does not occur, the cycle resets. Fertilization does not occur, the corpus luteum degenerates toward the end of the secretory phase, causing progesterone levels to fall. Spiral arteries kink and spasm, endometrial cells die and glands regress. Blood vessels fragment and the functional layer slews off, marking the start of a new menstrual cycle. This cyclical process is essential for female reproductive health. Estrogens promote oogenesis and follicle growth, exert anabolic effects on the reproductive tract, and induce secondary sex characteristics such as breast development, fat deposition, and pelvic changes. Estrogen also maintains low blood cholesterol and facilitates calcium uptake. Progesterone works with estrogen to regulate the uterine cycle, promotes changes in cervical mucus, inhibits uterine motility during pregnancy, and helps prepare breasts for lactation. These hormones are central to female health and reproductive function.